welcome to the Excel cycle. Today we will talk about cell cycle control and cancer. In this lesson, we will be talking about the cell cycle, specifically how it is regulated and what can happen if these controls malfunction. If you haven't already, go watch our video, an overview of the cell cycle, and then come back to learn about how it is controlled. We are going to talk about three main checkpoints, between G1 and S phase, G2 and mitosis, and between metaphase and anaphase. Let's start with the G1 S phase checkpoint, or the restriction point. This checkpoint is very important as it decides whether the cell will commit to division or not. The main function of this checkpoint is to ensure DNA is not damaged and can be readily replicated. Once the cell has passed this checkpoint, it has committed to completing the rest of cell division. First, we need to talk about cyclin-dependent kinases, or CDKs. Like all kinases, CDKs are an enzyme that will phosphorylate specific target proteins. The specific CDKs involved in the restriction point are CDK2 and CDK4. These CDKs are activated by the binding of cyclins, specifically E and D. The CDK4 cyclin D complex will phosphorylate the retinoblastoma protein, which usually inhibits DNA replication. After being phosphorylated, the RB protein is inhibited and can no longer stop DNA replication, allowing S phase to begin. Next, let's talk about the protein p53 and the role it plays in the checkpoint. If DNA is damaged, p53 will arrest the cell cycle in G1 by releasing CDK inhibitor proteins. These inhibitor proteins will bind cyclin CDK complexes and prevent the phosphorylation of RB, therefore preventing replication. p53 will then take on its second job. After pausing the cell cycle, p53 will attempt to repair the damaged DNA. If it is unable to do so, it will take on its third job and trigger apoptosis. Apoptosis is programmed cellular death. This occurs to prevent damaged DNA from being carried forward and passed to daughter cells. A mutation in the gene TP53, which codes for p53, is a common cause of cancer. This ruins the quality control of DNA and allows mutations to accumulate quickly and be passed on to subsequent daughter cells. This can cause uncontrolled cellular division or cancer. Next, let's move on to G2M checkpoint. The main function of this checkpoint is to ensure DNA has been successfully replicated before beginning mitosis. At this checkpoint, we again see the importance of CDK cyclin complexes, specifically cyclin B and CDK1. This complex will phosphorylate a variety of target substrates, causing cell cycle progression. It is important to note, in the case of damaged DNA, the cell will be arrested in G2 to await repairs. If these repairs do not occur, the cell will again go into apoptosis. Lastly, let's talk about the spindle checkpoint. This occurs at the transition point between metaphase and anaphase of mitosis. Because anaphase is an irreversible step, this checkpoint assures that sister chromatids are properly attached to spindle fibers before proceeding. It does this by scanning for chromosomes that are not lined up at the metaphase plate. If any of these chromosomes are found, anaphase will be put on pause until all chromosomes are aligned correctly. Let's go over what we learned today. At the restriction point, which occurs between G1 and S phase, DNA damage is assessed and repaired. We also employ cyclin-dependent kinases and cyclins to phosphorylate target proteins. At the G2M checkpoint, we ensure that DNA replication has successfully occurred. At the spindle checkpoint, we ensure that chromosomes are correctly aligned along the metaphase plate before they enter the irreversible process of anaphase. Errors in any of these steps can result in uncontrolled cellular growth or cancers. Thank you all for following along with today's lesson. Be sure to like and subscribe, as well as comment any topics you would like covered in future videos. Thank you.